Good afternoon, everyone, and welcome to the PreScouter lecture series. Today's webinar will cover synthetic biology, the future of pharmaceuticals, food, fuel, and chemicals. If you have any questions, we will be hosting an interactive question and answers session immediately following this presentation. This presentation is an abridged version of topics covered in the full report. An electronic version of the full report is available to you free of charge. If you do not receive a downloadable link to the report within 1 week, please email webinar@prescouter.com. Before we proceed, I would like to take a moment and acknowledge Dr. Patricia Rupert Nason, who's done an amazing job as the technical project manager on this presentation and report. Patricia attained her bachelor's degree in bioengineering at Rice University. She then went on to complete her PhD in chemical engineering from the University of Wisconsin at Madison, where she researched computational modeling to determine mechanisms for chemical reactions on catalytic surfaces. Patricia joined PreScouter in early 2014 as a scholar, working on a variety of projects over the course of 12 months. Patricia was then promoted to a full-time technical project manager in early 2015, now having completed 20 projects as manager, managing hundreds of scholars. And a brief introduction on myself. I am Ashish Basare, Chief Scientist at PreScouter. Prior to my time at PreScouter, I completed my PhD and postdoctoral fellowship at Northwestern University in mechanostereochemistry, nanomaterials, and organic chemistry. Prior to my PhD, I was a management consultant at one of those big firms just outside the Washington, D.C. Beltway, as well as having served in the United States Peace Corps in Nepal and performing cancer research at the National Institutes of Health. Okay, let's begin. The world is changing at an ever-growing rate. Many businesses struggle to stay abreast of relevant or even disruptive technologies, sometimes before it's too late. Two famous case studies include Rand McNally, a cartography company that was founded in 1856, while the Ottoman Empire was still a global powerhouse. Even though they were experts in their domain, Rand McNally failed to predict the importance of the global positioning satellite network being built out in the latter half of the 20th century. New companies that leveraged the new GPS technology, like Garmin, created a disruptive leap in technology so vast that Rand McNally was not able to innovate fast enough, and therefore Rand McNally was forced to file for bankruptcy in 2003. Another company that we probably all used growing up, Encyclopedia Britannica, suffered a similar fate. Encyclopedia Britannica was an educational institution for centuries, not decades. They were in business during the American Revolution. Through 2 world wars, they documented man's first steps on the moon and witnessed the rise of the internet. Sadly, the internet would be Encyclopedia Britannica's downfall in 2012, with Wikipedia leveraging the democratization of information to provide a completely disruptive approach to knowledge transfer. These 2 examples, as well as many others, illustrate how innovation is not linear, disruptive technologies are hard to identify, and competition for business is not always where you might expect it. Helping companies innovate intelligently is at the core of PreScouter's mission. We help companies like yours by providing answers to your most pressing questions, helping solve your innovation goals, and reducing your tactical work, allowing you to focus on strategy. To date, we have helped over 250 Fortune 500 size clients solve over 800 projects by working to answer questions about technology, connecting them to promising technologies, and identifying disruptive technologies before they become a threat. Allow me to briefly illustrate how the PreScouter process operates, as it's as simple as 1, 2, 3. First, you fill out a very simple statement of work, telling us a little about the criteria for the people, organizations, or technologies you would like to see profiled. Second, PreScouter assembles a team of scholars, usually recruited from Tier 1 research institutions globally. They are all under non-disclosure agreements with our clients, and these scholars use a combination of their own human intelligence networks as well as proprietary software developed at PreScouter to scan the entire globe based on methodologies employed by NASA and the Department of Defense. After several stage-gated meetings with you and your team, we'll present profiles that meet your criteria. You select the ones of most interest, and we identify questions that you would like on each profile. Through step 3, Through interviews and analysis, we'll build a report with enhanced profiles that answer your questions. We can even connect you to the interviewees. On average, our clients find that there is between 75 and 80% new and relevant information, which allows them to make informed decisions and, more importantly, take action. If you have any questions regarding how the PreScouter process operates, please feel free to contact me at any point. My email is ashish@prescouter.com. Now allow me to introduce the three talented scientists who have assembled the synthetic biology report and who will be today's speakers. Tusita Banerjee is currently a PhD candidate and graduate fellow in the Department of Chemistry at the University of Illinois Urbana-Champaign. Her research focuses on developing robust algorithms to study the dynamics of a small quantum system in a protein or chromophore backbone. She has developed a more realistic picture to model the quantum mechanical interaction in a redox reaction or a proton transfer reaction. These reactions are integral in understanding enzymatic complexes or photosynthetic reaction centers. She has received her bachelor's with honors from St. Xavier's College in Kolkata, India, and a master's in chemistry from the Indian Institute of Technology, Mumbai, India, ranking first in her class. To date, Tusita has completed 29 projects with PreScouter over the past 18 months. Our second speaker is Gurshagan Kandola, who is currently a PhD candidate in biological engineering at the University of Arkansas, Fayetteville. Her research focuses on the production of biofuels and bioproducts from cellulosic biomass and purification of bioactive compounds from natural extracts using advanced chromatographic techniques. She completed her bachelor's in biotechnology engineering from Punjab University, India, and a master's in food science and bioprocess engineering from the University of Illinois Urbana-Champaign. Gurshagan has completed 7 projects with PreScouter since she joined us in December of 2014. Our last scholar who contributed as an author to the report is Dr. Akila Vasan. Akila is a recent graduate with a PhD in food science from the University of Wisconsin at Madison. Her research focused on the survival of foodborne pathogens, Salmonella and Listeria monocytogenes, common in our foods. Her research experience, regulatory knowledge, and work in a pilot plant taught her to innovate and design solutions to scientific problems. She has completed 14 projects with PreScouter since she joined us just this past April. And without further ado, I will turn it over to Tusita, who will continue today's webinar. Tusita?
Synthetic biology can be thought of as an amalgamation of different fields. In essence, synthetic biologists are trying to gain inspiration from nature and build near-perfect systems that would be beneficial to the world. Researchers in synthetic biology belong to an interdisciplinary field with experts in genetics, systems biology, molecular biology, chemical engineering, electrical engineering, computer science, ethical and social sciences, and law. Synthetic biology refers to practices involved in engineering biology, and it could be to engineer protein and biomolecules and also to re-engineer whole systems. In engineering, people think of what something should eventually do and then build it based on its functionality. It is in many ways similar to electrical engineering, where to build the tools and techniques and then you modulate a process. From an electrical engineer's perspective, it can be thought of as working with an electrical circuit, except that the circuit is a cell and the components are biological switches. So the conditions definitely change. It can also be thought of from the perspective of design. The goal here is to make or build things rather than to study systems. We interact with biology every day, so how can we design systems that mimic biology? It is beyond genetic engineering. Yes, we need to know a bit of that, but we also need to expand our knowledge and work with researchers in other fields. In early years, researchers used to take snippets of DNA, know what each of the snippet meant, and aligned them in Microsoft Word. They could then cut, copy, paste these functions similar to editing a document, except that they were editing some genetic code. This was limited to what was in the file, and it was tedious and prone to error. Synthetic biology accelerated this process and added an computational layer of complexity to it to figure out what the function might be, rather than to just guesstimate. Soon, we'll have assembly line functionality where we can build complex functions. So, how do we go from building snippets of a plant to making microbial genes or life-saving drugs? Keeping all the information we have learned above, we can add another angle to this. Using computer science and engineering principles, we could think of the genes being different parts of database, each having its own functionality. We can then identify the parts, put them in the computer program, and build and model suggested networks with expected functionality. The individual functional components in the system can be the genetic components in a lab, which could be either frozen or otherwise, but it should be physically present. So we can learn to produce components, which of these would work and which wouldn't, and hence we can prevent waste. These are encoded to determine what is produced and more importantly, when things are produced. Having control on the when is the key difference in synthetic biology. So we basically turn the design to reality. In essence, we can think of synthetic biology as a combination of design, fabrication, and testing. Basically, DNA snippets can be considered as blueprint of life. Similar to connecting all the Lego blocks together, the DNA can be modeled to perform the required functions. This can be then tested in an actual living system such as a bacterial cell, which needs to perform a certain function under certain conditions, making sure that it is a controlled process at all times. Based on repeated testing, large-scale manufacturing, and tweaking of the product, innovation can occur. Keeping in mind the challenges the field might face, as with any other new and upcoming field, the role of social and ethical scientists, regulatory agents, as well as IP experts would be key in the controlled release of the synthetic biology products. Researchers are actively working in this area and will continue to do so to ensure a safe and sustainable world. Next, let's discuss the disruptive nature of synthetic biology in the pharmaceutical industry. But before moving to that, let's discuss why do we even need drugs? The World Health Organization defines health as the state of complete physical, mental, and social well-being, and not just merely the absence of disease or infirmity. Though much medical progress had led to considerable improvements in human well-being, still a huge potential for further improvements remain. The Disability Adjusted Life Years, or DALY, measures the years of life lost due to premature mortality plus the weighted number of years lived with disability or reduced well-being. The world tally for the year 2004 was calculated as 1.52 billion. With a global population of 6.44 billion, the average is around 0.24 years per person. Next, we consider the cost associated with diseases. The data shows that for the most costly ones in the US, in terms of total, which is direct and indirect costs, the top four categories of diseases account for 1 to 1.275 billion per year. So we need drugs. In spite of that, pharmaceutical industry's potential for improving the human health, it is affected by a twofold challenge with respect to innovation, with increasing costs and reduced returns. In the past 60 years, the pharmaceutical industry has seen major scientific and technological advances. Yet, this industry is faced with Ohm's law, which states that the number of US drugs approved per billion US dollar has halved every 9 years. Ohm's law corresponds to an average annual increase of cost of 8%. Since the mid-1960s, the process of drug approval has been modified to significantly improve the safety and efficacy of drugs for the use of general public. As a consequence of these scientific and regulatory changes, there has been an increase in the time taken and the cost of bringing a new drug to the market. So let's see how synthetic biology has tried to change this. Let's take the first case of Sitagliptin. Sitagliptin was manufactured using rhodium-catalyzed asymmetric enamine hydrogenation for large-scale manufacturing by Merck. The authors from Codexis applied a substrate walking, modeling, and mutation approach to create a transaminase with marginal activity for the synthesis of chiral amine. The active site of transaminase consists of a large and a small binding pockets as mapped on the structure of an acetophenone. A pro-sitagliptin ketone analog was designed to fit the large pocket for initial optimization of this part of the active site. After initial engineering of the large pocket, an enzyme variant was generated with activity of the desired substrate by excavating the small pocket. So let's look at the numbers. This synthetic bio route increased the overall yield by 10 to 13%. It increased the productivity by 53%. There was a reduction of 19% in the total waste. And for this, Codexis with Merck earned the 2010 Presidential Green Chemistry Challenge Award. Let's move to the next example of Cephalexin. Between 1975 and 85, Cephalexin was produced by a 10-step process with 30 to 40 kgs of waste generated per kg of product. In 1985, the waste to product ratio was reduced to 15. In 1995, the number of steps was reduced to 6 and the waste to product ratio was reduced to 10. In late 90s and early 2000, DSM and GIST Procase faced a lot of competition from companies in Asia and India on the market share. They then established the joint venture called ChemFirm. Both DSM and GIST Procase had expertise in complementary field. While GB had great depth in knowledge in microbiome and genetics, DSM could contribute its experience in biocatalysis and organic chemistry. Both of them used their knowledge to produce a one-step direct fermentation of adipoyl 7 ADCA. This product was then converted to Cephalexin via 2 enzymatic steps, replacing the traditional 13 steps of chemical processes. This improved the process and led to a total savings of 65% of energy and materials. It cut the cost by 50%. The waste was reduced to 1 kg per kg of product, and there was a decrease in the toxicity of waste. The original process required methylene chloride, silating agent, Dane salt, protected side chains, and acylating promoters. The new process mainly released aqueous waste streams containing harmless inorganic salts. Now let's move to the most successful case of synthetic biology, Artemisinin, which is also dubbed as Synbio's poster child. Malaria has around 300 to 500 million new infections per year. There are around 1 to 3 million deaths each year in areas of poverty. Who first endorsed artemisinin derivatives for treatment of malaria in 2004 and recommended it in 2005? At that point of time, artemisinin was produced from a plant called Artemisia annua. This was a very labor-intensive crop and it had a lengthy growing cycle. The time in between crop growth and artemisinin extraction was around 12 to 18 months. The commonly accepted artemisinin recovery yielded around 5 kg per 1000 kg of leaves and produced from 1 hectare of land. Based on this yield, it was calculated that around 1700 hectares of land are required to produce enough artemisinin to manufacture 100 million adult treatments per year. In 2004, only 4,700 hectares of land was available, and it was estimated that the total acreage could grow to 11,200 hectares. However, an additional source of artemisinin production was clearly required to meet the projections of a global demand of 400 million artemisinin treatments per year. Owing to the sensitivity of the crop to genetic backgrounds, cultivation conditions, and harvesting periods, the high cost of artemisinin itself, it was reported that it could be anywhere between 900 to 1600 kgs in 2006. Professor J. Kiesling's laboratory in California, Berkeley, and scientists at Amyris Biotechnology proposed the use of synthetic biology to develop strains for high-yielding biological production of artemisinic acid, which is a precursor of artemisinin. Earlier attempts, which had been made to produce this in a commercially relevant concentration, led to a production of only 1.6 grams per liter of artemisinic acid. However, by using a complete biosynthetic pathway which included the discovery of the plant dehydrogenase and a second cytochrome, which provided an efficient biosynthetic route to the production of artemisinic acid, they achieved 25 grams per liter of artemisinic acid. They also reported a well-developed, practical, efficient, and scalable chemical process for conversion of artemisinic acid to artemisinin using chemical source of singlet oxygen. To effectively steer these efforts and to maximize the project probability of success, One World Health, the Bill and Melinda Gates Foundation, worked with Amyris Biotechnology and provided a grant of $42.6 million. It was a three-way partnership between UC Berkeley, One World Health, and Amyris Biotechnology for product development. They determined the criteria to move the project forward and future partnerships needed to transition the research and laboratory scale development work to pilot scale and eventually manufacture it in a commercial scale. In addition, One World Health developed a commercialization strategy based on a thorough understanding of the worldwide regulatory requirements and on an analysis of the current artemisinin manufacturing supply chain and distribution model. In 2008, One World Health teamed up with Sanofi Aventis for commercialization of the project. Sanofi now uses the technology at large scale to produce artemisinin for treatments. More than 1.7 million treatments of Sanofi's Winthrop, that's the commercial name for artemisinin, was manufactured with semi-synthetic routes in Morocco and was shipped to the distribution center in Congo, Liberia, Nigeria, Niger, starting from August 2014. This public-private partnership of the Artemisinin project appears to be a perfect example of Bill Gates' concept of creative capitalism from his famous speech in the World Economic Forum. As he pointed out, the challenge was to design a system where market incentives would drive companies and scientists to do more for the poor.
Thank you to Sita for the nice introduction to the basics of synthetic biology and also for informing the audience how it's impacting the pharmaceutical industry. So now I will talk about synthetic biology and food. Every year, environmentalists measure the date on which we have used up all of Earth's resources for the year. It's called the Overshoot Day. With each passing year, without fail, this date falls earlier than the year before. We had it on the 14th of August in 2015 against the 19th of August in 2014. But the point here is that for the past 2 years now, we have been exhausting Earth's budget for the complete year in less than 8 months, which is more than alarming. According to World Health Organization, agriculture is going to face an unprecedented confluence of pressures, including a 30% increase in global population, intensifying competition for increasingly scarce land, water, and energy resources. In order to feed and sustain the world population by 2050, we need to increase the global food production from 8 billion tons to 13 billion tons. We absolutely do not have the natural resources to make such a huge leap so soon. The need is to do something better for our people and our planet while remaining profitable in our businesses. And this is only going to happen if we strongly consider alternate forms of food production. According to the Water Footprint Network, a Dutch non-profit research organization, the amount of water that's needed to grow protein-based food, which basically includes all kinds of meat, is way more than what's required to grow fruits and vegetables per pound of production. And this is especially thought provoking considering that a place like California, which has been in a state of drought for the past 4 years now, also happens to be the agricultural valley of the country, besides being home to a thriving dairy and meat industry. So, given the conditions as we see them, current animal and food production practices are clearly not sustainable. They are bound to lead to water shortages and drive food prices up. So the question is, do we have scientific techniques that can help make more food while making the whole system of food production more sustainable? The answer is yes, provided we are all open to trying. So has this been done before, or are we stepping into new territory? Again, the answer is yes, this has been done before, and it seems to be working pretty well. Let's travel back in time to understand how, in the early 1970s, one novel idea changed the complete dynamics of cheese production forever. So, cheese is produced using an enzyme complex called rennet that breaks down milk. Until a couple of decades ago, rennet used to be extracted by slicing a young cow's stomach. So, rennet used for cheese production was originally a byproduct of the wheel industry. However, as the demand for cheese increased, almost doubled from about 15 pounds in the 70s to 30 pounds in the 90s per person, and people wanted more Parmesan and cheddar, the wheel industry alone could not sustain cheese production anymore. Luckily, researchers were able to genetically modify microorganisms to artificially produce rennet with the same or perhaps even improved functional properties over naturally extracted rennet, thus revolutionizing the cheese industry. This product is called FPC, or fermentation produced chymosin, chymosin being the key component of rennet. In 2008, approximately 80% to 90% of commercially available cheeses in the US and UK were actually made using FPC. So, the demand for cheese was now met in a much more sustainable way, and also due to FPC's non-animal origin, cheese became available for consumption to vegetarians. This is one of the most crucial examples of synthetic biology in food. So that was 25 years ago. Now, let's look at a few examples where synthetic biology is currently making headlines in the food industry. The smell of vanilla when you cut open a pod is priceless, but how is vanilla produced and what makes the flavor so unique? Natural vanilla, indigenous to the Madagascar Islands, is a complex mixture of over 200 compounds. The age-old process of obtaining natural vanilla from cured pods is laborious, expensive, and insufficient to meet the demands of the world. In 2010, the global demand for vanilla was estimated at around 16,000 tons, and even today, 99% of this demand is being met by chemical synthesis of vanilla. in which case we call it vanillin, the term for fake or imitation vanilla. Since the past few years, there's tremendous research going on to make more environmentally friendly routes of vanillin synthesis a reality. But there's one company that has revolutionized this process using synthetic biology techniques, and that's Evolva. Evolva uses yeast to produce vanillin, the same organism that we use for fermentation of beer and baking of bread. Yeast is genetically modified to contain genes that can transform sugar into vanillin during fermentation, and once the process is complete, the modified yeast is removed, leaving no traces behind in the final product. So you don't have to worry about the safety of yeast brewed vanillin. In fact, this is a more sustainable method of vanillin production, better than both the natural and synthetic routes. Coming back to the fact that the production of beef, pork, and chicken has such a huge water footprint, researchers are now developing alternate methods to produce meat. A study done in 2011 by Oxford University and the University of Amsterdam found that meat cultured in laboratory produces 95% less greenhouse gases and consumes roughly 90% less water when compared with the meat that's produced by commercial livestock production methodologies. So, the general idea is to extract stem cells from the animal or the bird, like chicken, cow, or pig, and allow these cells to proliferate in a bioreactor under controlled conditions of temperature, humidity, sterility, and the right substrates that ensure this perfect environment for cells to grow and amplify. Electrical, mechanical, and biochemical stimulation results in proliferation of cells, ultimately leading to the development of muscle tissue. This is actually a classic example of synthetic biology that does not involve any genetic engineering. Genetic engineering can be thought of as a subset of synthetic biology, even though the words are used interchangeably most of the times, but they don't mean the same thing. In this case, yes, we are synthesizing cells in laboratory, but we are not playing with their genetic content, so the final meat product would not be GMO. Back in 2008, PETA had offered a prize of $1 million to the first company that would bring laboratory grown meat to consumers by 2012. Unfortunately, nobody won the prize because the parameters are in fact very stringent. You have to produce meat in commercially viable quantities, it has to have good texture and taste, so it's definitely very challenging and it will take at least a decade to make the product cost competitive with traditionally sourced meat. But as of 2012, there are 30 labs from across the world that are working on in vitro meat research, and there's an Israeli lab that's taking this one step forward by trying to make a whole chicken out of a single cell. Not just the muscle tissue, but the whole chicken. So that would definitely be one breakthrough to watch out for. Next, talking about food crops. Across the world, and especially in parts of Asia, rice is a staple source of carbohydrates. It's eaten every day in every meal in many households. And the reason this information is important is because, believe it or not, but rice cultivation is a major source of atmospheric methane. Up to 100 million tons of methane produced annually comes from rice cultivation. After carbon dioxide, methane is the most harmful greenhouse gas and contributes to 20% of the world's global warming. Considering that majority of the world's population consumes rice, the need for sustainable rice production is even more pressing. Swedish researchers have produced genetically modified rice called the Susiba rice that is higher in its starch content and has significantly lower methane production. A simple solution to the problem achieved by correction of carbon flux, that is, by diverting more carbon into the grains and stems and less into the roots. The amount of carbon that's going into soil decreases. Decomposition of carbon by microbes that are present in the soil is what leads to methane production and is successfully averted by Suceba rice. So, having talked about food, now I will talk about how synthetic biology is proving disruptive in the field of fuels and chemicals. I think research has already established, very well established, the fact that growing population, depleting petroleum reserves, and increasing greenhouse emissions are having a profound impact on climate. The deleterious effects of this more than a century old fossil fuel based economy are not new to us. Thankfully, there is a small research community in academia and industry that is committed to transitioning to a more sustainable bio based economy. And in the next few slides, I will show you how synthetic biology is at the very core of making this possible. Taking a few examples of commonly used chemicals first. Acrylic is a major industrial chemical used in a wide range of products such as paints, diapers, and detergents. Petroleum derived acrylic is currently a $10 billion industry. A company named OPX Biotechnologies has successfully produced biologically derived acrylic using its proprietary technology called EDGE, that is, efficiency directed genome engineering. Now, in this schematic, the green color represents the laboratory phase of development and testing, and the blue color represents production at plant scale. So we start with millions of natural strains of the microbe, which are first genetically modified and are then experimentally evaluated to see the impact of genetic modification on performance. This data is then used to design a strain with the right combination of genes that gives the highest possible output of acrylic. You optimize the microbe and the growing conditions, produce acrylic at pilot scale, recover your product, and if the numbers turn out to be economical, the process is scaled up to commercial scale. So this is how the process generally looks like, be it for the production of acrylic or any other chemicals such as succinic acid, adipic acid, or even biofuels. The company received private funding of $36.5 million in 2011 and to develop this product, and because of the great progress made by them towards product commercialization, OPX Bio was acquired by Cargill in April this year. Another major commodity chemical is isoprene, the monomeric unit that's polymerized to make rubber, which is further used to make thousands of different products, but tire production is by far the largest end use of rubber. Almost 70% of rubber goes into tire production according to 2013 world statistics. Up until now, rubber has either been derived naturally from trees or synthetically from petroleum. The collaborative research agreement between DuPont and Goodyear in 2008 to make bioisoprene from renewable raw materials came as a major breakthrough to make the rubber and tire industry less dependent on these conventional processes. They have developed a fermentation process where genetically engineered microbes that have been optimized to produce the enzyme isoprene synthase are used to convert glucose into isoprene. So instead of deriving isoprene from trees or petroleum, both of which are limited resources in their own ways, we can now produce isoprene in fermentation tanks using synthetic microbes. The first demonstration tires made with bioisoprene technology were showcased at the United Nations Climate Change Conference in Copenhagen in 2009, and as per Biofuels Digest 2014, commercial production is expected in 2016. Biodegradable plastics have drawn increasing attention in the past decade due to obvious reasons, due to their environmentally friendly properties. And after 20 years of rigorous research, Metabolics has successfully commercialized its microbial fermentation process where natural sugars are transformed into biodegradable plastic. The product is currently being marketed by the company as Mirel Bioplastics. As of 2014, Metabolics was producing 50,000 pounds of PHA biopolymer per month at its pilot facility, and it plans to continue to expand its production capabilities. Another joint venture that's worth mentioning here is the collaboration between DuPont and Tate & Lyle that started back in 2004 to produce the multi-purpose chemical 1,3-propanediol from corn sugar. The global 1,3-PDO market is estimated to grow from roughly $300 million in 2014 to about $600 million in 2020. This research project earned the US Presidential Green Chemistry Award in 2003. They expanded their plant capacity by 35% in 2011, and with their latest partnering with companies from France and South Korea, this technology is set to expand in Europe and Asia as well. BioPDO is currently being marketed as Zemia, that can be used as a functional ingredient in food and beverage products, cosmetic and personal care products, and as Sustera that has industrial fluid applications. Next, talking about fuels. So, first generation biofuels are produced by the action of microorganisms and enzymes through the fermentation of sugars or starches that are present in food crops such as corn, wheat, and sugarcane. ADM is the biggest bioethanol producer in the US, churning out almost 1.75 billion gallons per year. But the corn based ethanol model in the US and even the sugarcane based ethanol model in Brazil have both achieved a kind of technological maturity. And from a food versus fuel standpoint, also, there's only a certain percentage of crop that's mandated for fuel production, and you can't really go beyond that level. This led to a shift in focus to second generation biofuels, which are also produced by the action of microorganisms and enzymes, but through the fermentation of lignocellulosic biomass such as switchgrass and miscanthus, or the non-food part of crops such as rice and wheat straw, corn stover, and even woody biomass. Now, compared to starch, it's much more difficult to break down cellulose and lignin, and that's where new generation synthetic biology comes into play. Robust industrial strains obtained by screening millions of genetically engineered microbial strains and highly efficient synthetic enzymes are used to degrade cellulose. Companies such as Novozymes and Codexes are at the forefront of developing such enzyme cocktails. DSM's expertise in producing yeast and enzymes that can co-ferment complex sugars, and POET's expertise in feedstock logistics and grain ethanol production gave rise to a joint advanced biofuels venture, and their commercial-scale cellulosic ethanol plant was the first of its kind to open in the US last year. Their proprietary technology is now available for licensing to third parties. Another company that was able to commercialize the cellulosic ethanol model is DuPont, and their plant is also up and running in Iowa. Third generation biofuels or advanced biofuels are produced from algae and other microbial species called cyanobacteria. Advantages of algae and cyanobacteria are that these microbes use sunlight, carbon dioxide, and wastewater to grow. So all the raw materials that are required to grow algae are freely available. Compared to yeast that's used in the production of first and second generation biofuels, the genetics of algae is far simpler and easier to manipulate. Algal biofuels research has received a fair amount of government as well as industrial funding in the past few years. about $45 million by US Department of Energy, and a staggering $600 million has been given by the fuel giant ExxonMobil to Synthetic Genomics, the company that constructed the world's first synthetic cell. Even though algal fuel is struggling with the challenges of throughput and production economics when compared with the cheaply available gasoline, yet startup companies such as Sapphire Energy and Solazyme are transforming algae into products ranging from biodiesel and jet fuel to protein-based food ingredients and oils for personal care products, once again by investing in synthetic biology research at the front end of the process. Founded in 2003, Solazyme had surpassed $76 million worth of funding in 2009, and it has now entered into key partnerships with the chemical giant BASF, the food company Bunge, and the cosmetic company Sephora to diversify its distribution channels. Founded in 2007, Sapphire Energy is in fact a product of industry-academia collaboration, and it received $144 million worth of investment in 2012. It is now a major algal biofuel company in the US. Due to their extensive research efforts and commercial success, both the companies have consistently been listed in the list of 50 hottest bioenergy companies by Biofuels Digest. Having talked about some of the conventional areas of application, over here in this slide I will talk about bioremediation, an area where synthetic biology holds immense potential in the future. The iGEM Foundation is dedicated to the advancement of the field of synthetic biology. Their main program is the iGEM competition, where undergraduate student teams from all over the world compete against each other. They work with a kit of biological parts provided by iGEM to build and test biological systems in living cells ranging from bacteria to mammalian cells. In 2012, one of the finalist teams from University College London engineered bacteria, E. coli in particular, for enhanced adhesive properties aimed to aggregate tons of microplastic pollutants in marine environments to create the so-called plastic islands, which can later be recovered for bulk treatment. Considering that millions of tons of plastic waste is floating in the North Pacific Gyre, innovative ideas such as these are slowly making synthetic biology an extremely handy technology to tackle the most difficult problems we are facing as a humanity today. So, from pharmaceuticals to food to fuels and chemicals, we have seen how synthetic biology is starting to make an impact in many areas of our daily lives and businesses. It truly has the potential to be a major disruptive force in the 21st century. We hope you enjoyed the presentation and looking forward to your questions in the live Q&A round. Thank you.
Thank you very much to Sita and Gershagen for your kind and considerate presentation. We have gotten a couple of questions both in the chat and offline. The first question that I have is for Gershagen, and Neil Klein asked, how is the global market of synthetic biology going to look 5 years from now? Gershagen?
So by 2020, it is projected that this market is going to increase up to about $17 billion US, and it is going to have maximum commercial applications in the fields of medicine, energy, environment, agriculture, and food.
All right, another question we have was on what areas this is going to have the biggest impact on geographically. Tusita, do you have the answer to that?
I predict that it would be around the Asia Pacific because the market there is growing at the highest rate. So that would be my answer.
All right, and last of all, we heard a question from Jessica Crummy, it looks like, and that she wanted to know what was the most promising areas we felt for synthetic biology. Gershigan, would you like to take that question?
Yes, so pretty much the 4 areas that we've touched upon today in the webinar: pharma, chemicals, food, and fuel. Out of these, I would still say pharma and chemicals are more promising than fuel and food, just because medicines and chemicals are still low volume and high value end products. And food and fuel are, the conventional sources of food and fuel are still cheaply available. So at least for the next 5 or 6 years, these are the 2 fields, pharma and chemicals, which should be the focus of synthetic biology if you want to see more return on your investment.
All right, if anyone else has any questions, please do drop them in the chat. Otherwise, it looks like we are just about at the top of the hour. So if I don't see any more questions coming right up, I think we'll call this an hour. Thank you so much for taking the time to join us today, and I hope that you have a wonderful afternoon.